Hi, everybody. We're back. Part two. We're going to switch gears a little bit from bacteria and move on to viruses. So, viruses are very unique in that we study them in biology, even though they're technically not living organisms. They have a ton of characteristics of life, including the genetic material, including their ability to evolve. So, clearly, these are things that are so tightly interwound with living things that we cannot have a proper biology class without talking about them. And of course, we're going to talk about them even more in the middle of a pandemic. So, structure of viruses. They are incredibly small. So, it took a very long time to actually understand of their existence because no one could actually get any images of them until the middle of the 20th century. They were very poorly understood. They often are smaller than even a ribosome. They have varying genomes, anywhere from 4 to 400 genes. 400 is a lot. Most of them are much simpler than that. And their genomes come in various forms. That shorthand there stands for double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. And we can find all of them. Typically, viruses have some sort of protection on the outside. And we call that protective layer of proteins a capsid. Those capsids are particularly important and something we focus on in biology a lot because often the proteins on that surface are what help the immune system detect that virus. So if we can get capsid proteins and present them to our immune system, we can sometimes get our immune system to respond. And if we are able to do it in a way in which the immune system is responding to a virus protein without in fact actually responding to the virus. In other words, we give a person the proteins without the rest of the virus, then we might be able to help a person establish some immunity. So this is what an immunization is. So we are obviously working on these types of things for the current virus epidemic. So we also have viruses that have envelopes, which are membranes. And that is a common one, especially among viruses that infect the lungs, which includes flu, and yes, of course, COVID-19. So, moving on, here we see some images and diagrams of some varying types of viruses out there. This one may look a little bit familiar. This is a little bit, we saw this in our Hershey-Chase experiment, one of the bacteriophages. This is a flu virus that is covered in that membrane. Here is an adenovirus. Here is a tobacco mosaic virus. That's one that affects plants, not humans. So, general stuff about a virus. They are parasites of the ultimate sense. They cannot live without a host. So, most viruses have a host range. In other words, they can infect a specific set of organisms. Some have very broad, and probably the best example I could come up with a virus with a broad host range is rabies. The rabies virus can infect every mammal that we at least think so. Obviously, we probably don't have cases in every mammal, but we think that the rabies virus could infect that. I mean, for example, I don't know if there's ever been a case of a dolphin getting rabies, but again, how does a dolphin get rabies? I mean, there's no rabid raccoons running out into the surf and biting a dolphin. So, I mean, that would be a crazy image if you can imagine that. So, rabies, we believe, can infect all mammals and it affects the brain. Some viruses have a very narrow host range, like HIV, can only infect humans. There that virus will not even infect our closest relatives. In fact, you could argue that HIV has even more of a specific host range in that it doesn't infect all human cells. It actually targets some very specific cells. So it has a very, very narrow host range. And viruses only reproduce inside host cells. They cannot reproduce outside. They generally do not last long outside of their host. Usually from minutes to days is the longest amount of time. There are a few rare exceptions under the right conditions, but typically speaking, viruses do not last long. So what's the general strategy of a virus? Basically, the virus, there is a viral particle, and that particle has to come in contact with the host cell. The viral particle does not sense the host cell and does not go towards it. It just simply bumps into it, which is why there needs to be millions and millions of these viral particles in order to get the host to actually get an infection, because it's random. And once the viral particle comes in contact with the host cell, the genetic material of the virus enters the host cell, and then slowly but surely, or sometimes in a very fast way, the virus actually takes over the cell and uses the machinery that is already in the cell to make new viral proteins and new viral genetic material. And typically then the virus will self-assemble inside the cell and either destroy the cell to be let out or go through a mechanism in which it goes through exocytosis, and the new virus exits the cell that way. But typically, for whatever happens, the virus taking over the cell is not good for that cell. This is an example of a typical viral infection. The virus comes in, lands on the cell. The DNA, this is a DNA virus. The DNA will then take over and it starts producing it copies the genetic material. It also then makes messenger RNA and transcribes and then translates that, makes proteins. Now you have the proteins and the DNA inside the cell. They self-assemble and then often exit the cell, in sometimes a very dramatic fashion in which the cells actually burst and the cells actually are destroyed in the process. So, two types of cycles. We have what's called the lytic and lysogenic cycle. Lytic and lysogenic cycles are cycles that have varying effectiveness, and especially how they're employed. But lytic cycle is seemingly the worst, but in some cases maybe not. The lytic cycle, basically, the virus quickly takes over the cell, destroys the host cell's DNA, and then becomes a virus factory. So the cell, even though it might be a human cell or another organism cell, it no longer functions with any sense of being a human cell. It is totally 100% a virus cell at that point, if there is such a thing. It has been fully taken over. The lysogenic is a little bit more tricky, where the virus actually enters the cell and then hides its genetic information into the host's genetic information. And you actually have this, it's more common in bacteria, but it can happen in other organisms, where you have a bacteria that will become what's called a prophage. Prophage is essentially a bacteria that has virus inside of it, but it doesn't know it. And it's every time it's reproducing the bacteria, it's also reproducing the viral DNA. And it lets that go on for generation after generation until all of a sudden there's a switch where you can actually trigger that viral DNA to start being produced, and you actually produce some of that. We believe that a bunch of human DNA is leftover viral DNA that's actually sort of gotten stuck, and that switch to trigger it to come back on has never occurred. So we have a lot of junk DNA that's actually left over from viruses of many, many generations ago. Here's an example of the lytic cycle. Virus comes in, takes over the DNA of the host cell, becomes a factory. We have a cell that's chock full of virus, and then the cell explodes. If you've ever seen, there's a book called The Hot Zone. I recommend it to anyone, although it's a scary book, and especially at this time. But it focuses mainly on Ebola, which is a really, really nasty virus. But it has a description in which it talks about an autopsy of a person that was infected by a virus very similar to Ebola called Marburg. And when they do the autopsy, they find that most of the organs inside this person are really not distinguishable because so many cells have been destroyed in this way. And they basically all the cellular structure is gone because so many cells have been destroyed. And it also finds these black specks, and those black specks are about 1 million of these particles all chunked together in one big gigantic clump. This is an example of a lysogenic. See, what we see here is that, again, a virus comes in, enters the DNA, and what the trick here is now we take this DNA, we enter the host cell DNA, and now the host cell can actually continually replicate, and it can stay in this lysogenic cycle for a long, long time until something triggers it, where you actually trigger it back and go into the lytic cycle. Okay? Again, this one is obviously destroys the cells the most, but this one kind of allows the virus to hide for quite a significant amount of time. Okay. Animal viruses, they come in all shapes and sizes. A lot of them have cell membranes. And the cell membrane proteins, those viruses that have those cell membranes, they don't actually have to destroy the cell. They can actually use the process of exocytosis to produce new virus particles. So it doesn't actually kill the host cells. And a lot of viruses are these things called retroviruses. They're made of RNA. And the reason they're called retro is because they go backwards. In humans, we never go from RNA back to DNA, but viruses do. So they use a chemical known as reverse transcriptase, and it turns the RNA back into DNA. Now, the best example of a retrovirus that you know of is HIV. And it's where we try to fight HIV is because we try to block that process of RNA turning back into DNA because it doesn't happen in any of your cells normally. Unfortunately, coronavirus is not a retrovirus. It is an RNA virus, but it never turns its RNA into DNA. It just turns its RNA into more RNA. It replicates RNA. So those are one of the problems with retroviruses and all RNA viruses is that they actually evolve rapidly. This is a list of viruses, and you can see there's a lot on here, and this is certainly by no means a full set, but you can see some of them are double-stranded DNA, single-stranded DNA. You can see coronavirus down here, and this list was made long before our current coronavirus, but this is using the previous example known as SARS, and lots others out there. This is actually a nice example of HIV. This HIV is also, it's a membrane-bound virus. It enters the host cell. It actually turns that RNA back into DNA, and that DNA actually incorporates itself. This is one of the reasons why HIV is so hard to fight, is because it actually incorporates itself inside these cells. These cells actually don't behave as if they're sick. And so it's so difficult for your immune system. And guess what these host cells are? They're your T cells. And your T cells help fight off viruses. So it attacks from the inside. That is one of the really unique features of HIV. I do have more on the coronavirus, but I'm going to save that for a bonus video because I don't want to sell it short and I'm running out of time.